Hello students, I am Amruta from Biotechnology Department of KTHM College Nasik. Today we are going to discuss one topic from our immunology course, that is antigen and antibody interactions. So before going to our topic, first we will see few basic concepts. First one is the antigen molecule. So what is antigen? It is basically any foreign particle that enters your body and your immune system recognizes it as a foreign and mounts an immune response against it. So there are two concepts that you need to understand here. First one is the antigen and second one is the immunogen. Now, antigen can be a particle that can combine with antibody molecule. But if you consider immunogen, then what is it? It can activate the immune response. So, all the immunogens, that means all the molecules or particles which can activate the immune system, can bind to the final product of the immune response, that is antibodies. But not all the antigens can activate the immune system. For example, there are haptens which are so smaller in size that they are not capable of activating the immune response, but they can surely bind to the antibody molecules. Then second concept is the antibody. Now what do you mean by antibody? So antibody are also known as immunoglobulins. They are the protein molecules that can recognize this antigen that has entered your body, and they can mount an immune response or activate the immune system to eliminate that antigen from your body. But for this, for activating the immune response, what is required? There should be an interaction between these antigens and the antibody molecules. But how they interact? These two, now if you consider, then they are the bulkier molecules. So whole molecule of antigen cannot interact with the antibody. So there is a specific site or specific location present on the antigen which is called as epitope or antigenic determinant, which can get attached to the antibody molecule. On the other side, antibody molecule as a whole is not going to interact with antigen. So on antibody also there is a specific site located which is called as antigen binding site, and that in that antigen binding site there are specific residues which are going to interact with the antigenic determinant. That site is called as paratope. So epitope is present on antigen, which is also called as antigenic determinant, and paratope is present on the antibody. And these two, epitope and paratope, are going to interact with each other to form the immune complex. So similar to enzyme and substrate complex, here when antigen and antibody combine with each other, the complex which is formed is called as immune complex. So these are the few basic concepts. Now we will move to our today's topics. So in today's lecture, what we are going to discuss is first, in which are the non-covalent interactions or non-covalent forces involved in this antigen and antibody interaction. Then we will see some basic characteristics of this antigen and antibody interaction, like what do you mean by specificity, affinity, avidity, and cross-reactivity. Then we will see one basic concept, that is Lattice hypothesis, and then we will see what do you mean by the zone phenomenon when you consider a precipitin curve. So the objectives of this today's session is, firstly you need to understand the concept of antigen and antibody that just now we have discussed. Then second one is you need to understand what is the significance of this antigen and antibody interaction. Like as I have told you, antigen is foreign to your body. So when it will enter inside your body, if it is a pathogen, for example bacteria or virus, then it is going to be harmful to your body. That means what it will do, it will enter your body and it will start damaging your own cells or own tissues. So in this case, if the antibodies are secreted against this antigen, then what these antibodies will do is they will recognize this antigen, they will go and get attached to this antigen molecule, and they will activate the immune system. That means they will send a signal to the immune system that this molecule should not be present in your body and it should be eliminated from your body immediately. So when this is possible, when antigen can be eliminated, if the interaction between antigen and antibody occurs, then only this elimination of antigen is possible. So this is the significance of these interactions. Then we will see which are the non-covalent forces. That means The non-covalent forces, basically in this, what do you mean is, these are the interactions in which no electron sharing is involved, like the covalent bond. Okay, covalent bond means a bond which is formed by sharing of the electrons. Opposite to that, in non-covalent forces, no electron sharing occurs. So there are few types of these non-covalent interactions. These are basically weaker interactions, but they help in attachment or interaction of this antigen with the antibody. Then we will see few characteristics of these interactions, and lastly we are going to see what is lattice hypothesis and the zone phenomenon. So first topic is the forces involved in antigen and antibody interaction. So as I have told you just now, when antigen and antibody interacts with each other, they are going to result in the formation of immune complexes. So in the formation of immune complex, what basically happens is, similar to lock and key, this antigen will go and get attached to the antigen binding site which is present in the antibody. So if this lock and key are specific to each other, then only lock is going to open. So similarly here, if antigen and antibody are having complementary structures to each other, then only antigen can get attached on the antigen binding site on antibody molecule. So what should be there? There should be a good fit between the antigenic determinant which is present on antigen molecule, that is epitope, and the antibody combining site, that is paratope, present on the antibody. So this good fit is going to determine whether the interaction between these two will be stable or not. So to make this interaction stable, there are few attractive forces which are involved. First one is electrostatic bond, second one is the hydrogen bonding, third one is van der Waals interactions, and fourth one is the hydrophobic interactions. So one by one now we will discuss all these interactions and what do they mean, but basic thing will remain same, that is, these all are the types of the non-covalent interactions. So first interaction is the electrostatic bond. Now what do you mean by this bond is, if there are two oppositely charged molecules present, like if one molecule is carrying a positive charge and second molecule is carrying a negative charge, then what will happen? Due to this opposite charge which is present on the molecules, they will get attracted to each other, similar to magnet. Okay, in magnet what happens is, if opposite poles are present, then they will get attracted to each other. If same poles are present, then they will repel each other. So same is the principle here also. So example is, you can see this is an antibody molecule. Now on this antibody molecule, in paratope, that is in the antigen combining site, there are amino groups present. So they are imparting the positive charge on this antibody molecule. And second is the antigen molecule, which is having aspartate residue on its surface, and due to that it is carrying negative charge bearing group, that is carboxyl group, on its surface. So what has happened here? Here positive and negative groups have come closer together, and due to that this antigen and antibody came together to form the immune complex. So this is the meaning of electrostatic bond. Then second type of interaction which is present is the hydrogen bonding. Now what do you mean by hydrogen bonding? If you see this water molecule, then what you can see is, there is oxygen atom present, and to that hydrogen is attached. Now oxygen is electronegative atom, that means it has tendency to attract the shared pair of electrons towards itself. So arrow indicates that the electrons are attracted towards the oxygen. Now if an atom is accepting the electrons, then it is going to bear a negative charge. So you can see partial negative charge is developed on oxygen, and on the other hand, from hydrogen, electrons are pulled away. So basically it is giving its electrons, so it is bearing partial positive charge. Now due to this partial positive and negative charges which are formed, what will happen is, hydrogen atom of the first water molecule will get attracted to the oxygen atom which is present in the second water molecule. So this type of bond, where hydrogen is located between the two electronegative atoms, is called as hydrogen bonding. So you can see in the figure there are two water molecules in which hydrogen is located between the two oxygen molecules which are carrying the negative charges and hydrogen is positively charged. So same principle of the electrostatic bond will apply here also. Due to the partial opposite charges which are present, these atoms will get attracted to each other. So it is basically a type of electrostatic interaction that involves hydrogen atom which is located between the between a pair of electronegative atoms. Then third type of interaction is Van der Waals interaction. Now, what do you mean by this? So, these forces actually depend upon the interactions between the electron clouds. Now, we all know the structure of atoms, and in atom, we know that there are neutrons, protons, and electrons present. Inside the nucleus of atom, like you can see on screen, there are two atoms given. Inside the nucleus, there are neutrons present which do not have any charge, so they are not shown in this figure, but there are protons shown. So you can see in the nucleus of the atom, that is in the center, that there are plus charges given. Why these plus charges are present? Because there are protons present inside the nucleus which impart positive charge to the nucleus of an atom. On the other side, where electrons are present, they revolve around the nucleus in the orbital. So these are going to impart negative charges on the atom, and as a result of this positive and negative charge, whole atom is considered neutral. But what happens now in this van der Waals interaction is, when these two molecules will come close to each other, then what will happen is plus charge which is present inside the nucleus will attract the negatively charged electrons on the second atom, as you can show in the figure, and due to that partial polarization will take place. This is called as fluctuating dipole which is formed, and due to this fluctuating dipole formation, what will happen is these two atoms will be attracted to each other. So again, basic principle, that is plus and minus are going to attract each other, is going to remain the same. But when it is going to occur, if two atoms come close enough so that their electron cloud should interfere with each other. So these forces depend upon the interaction between electron clouds that surround the antigen and antibody molecules. Then fourth type of interaction is the hydrophobic interaction. Now what happens in this is, there are some hydrophobic groups or side chains present like valine, leucine and phenylalanine amino acid side chains. So what happens is, these side chains tend to associate, that means they tend to come close together due to the van der Waals bonding just now we have discussed, and due to that what happens is, in the aqueous environment, this hydrophobic groups, hydrophobic means what? They do not like water molecules, and if water is present around them, then they will try to run away from the water molecules. That is the meaning of hydrophobic, that is water fearing. So in aqueous environment, what will happen? These hydrophobic groups will try to run away from the water molecule, and during this what will happen? They will come close together also due to the van der Waals interaction. So as a result of this, what will happen? There is a paratope, that is antigen binding site present on the antibody molecule, and this is the antigen. So if water molecule is present between them, then when they will come close together, this water molecule will exit from the binding site. So this is the result of hydrophobic interaction, and due to this antigen and antibody will interact with each other. So as a consequence of this, the distance between the antigen and antibody will decrease, and it will enhance the energies of attraction which are involved between them. So these are the four forces which are involved in the antigen and antibody interaction. Now it is not possible that only one force will act in the antigen and antibody binding. All the four types of forces are involved, and together they are going to contribute to the attraction between antigen and antibody and their binding to each other. So when this antigen and antibody interact, what are their general characteristics of the binding? So first one is the antibody specificity, then second is the affinity of antibody for the antigen, third is the avidity, and fourth one is the cross reaction. So one by one now we will discuss what this means. So first one is the antibody specificity. Now when you consider the immune response, then it is very specific. For example, if a specific pathogen is entering your body, say influenza virus is entering your body, and if antibodies are secreted against this influenza virus. But after few days you are getting infection of COVID or coronavirus. So it is not possible that antibodies which were secreted against the influenza virus will help you in eliminating the coronavirus also. Why this is so? Because the antigen binding site which is present on the antibody is very specific for the antigen. So each antibody protein can bind specifically with unique epitope present on the antigen only. And how this is possible or why this is possible? Because see the structure of the antibody molecules, it is made up of constant and variable region. The region of antibody structure that you can see in the purple color is the constant region. But the region which is shown in the green color is the variable region, means what? Their amino acid sequence can alter to generate variety of structures that can fit different types of antigen inside it. So this allows the flexibility in the structure of antibody molecule, and due to that it can generate number of structures which can recognize number of antigen molecules. So you can see that in the variable region there is antigen binding site present in the antibody structure. Now due to this what happens is, now see here, this specificity is determined by the overall degree of complementarity between antigenic determinant and antibody binding site. Means what? You can see in this diagram there is a representation of antibody given. Now this A antigen is not specific for the antibody. So what happened just now? That A came and it was not getting attached to the antibody and it was released immediately. On the other side you can see antigen B is carrying an antigenic determinant on epitope on its surface which is complementary to the antibody structure, and due to that it came and remained attached to the antibody structure. So this, why this is so? Because B is specific for the antibody and A is not specific for the antibody. So differences in this degree of complementarity will determine the affinity of antigen and antibody reaction. Now, what is the affinity? So when you consider a specific antigen and antibody interaction, then you need to determine what is the strength of the reaction. That means whether that interaction is stronger or weaker. So see for example, this is one antigen binding site present on the antibody and this is one antigen. So what will happen when they are interacting with each other? Either this bond can be stronger, that is it cannot be, this antigen cannot be removed easily, or this bond will be weaker, so this antigen can be easily released. So the strength of this bond formation is called as affinity. What I am saying here, single epitope present on the antigen is interacting with single antigen binding site present on the antibody. So the strength of interaction of single epitope and single antigen binding site is called as affinity. Okay. Now if this affinity is low, that antigen is going to bind weakly and it can dissociate readily. But if this strength or affinity is higher, then what will happen? This antigen will bind more tightly and it will remain bound for the longer duration. Now in this affinity there are two forces involved. One is attractive forces and second one is the repulsive forces. So as a result of these two type of forces, the strength of interaction is going to vary. Now second term is the avidity. Now again this term signifies the strength of the interaction between antigen and antibody molecule, but still it is going to differ. How? See, in affinity what I have told you, that single epitope and single antibody, single antigen binding site on antibody was interacting with each other. But when there are multiple epitopes present on the antigen and there are multiple antigen binding sites present on the antibody, then what will happen? When first bond is forming, then this second epitope and second antigen binding site are coming closer to each other, and due to that the probability of their attachment is increasing. So as a result what will happen is there will be multiple interactions between antigen and antibody. So due to this what is happening here is, if multiple interactions are there, then it is not easy to break these interactions. So the strength of total interaction is going to be higher as compared to affinity, that is the strength of individual reaction. So what is the difference between affinity and avidity? Affinity determines the strength of individual interaction between the single epitope and single antigen binding site on antibody, and on the other hand there are polyvalent antigens present and polyvalent antibodies present. So when multiple interactions are there, then total strength of all these interactions together is termed as avidity. So affinity at one binding site does not always reflect the true strength of antigen and antibody interaction. The strength of these multiple interactions between multivalent antibody and antigen is known as avidity. So if affinity is low, but due to multiple interactions avidity is increasing, then antibody can efficiently bind to the antigen. So we will see one example. On screen you can see immunoglobulin G is present. Now this antibody has two antigen binding sites. So if it gets attached to the first antigen, then it will give, then what will happen? Second antigen will also come closer to the antibody, and the chances of the interaction at second site is going to increase. So this happens with the IgG, which has two antigen binding sites. So this is the IgM antibody. Now if you consider IgM, then it is carrying the 10 antigen binding sites. So what will happen? If you compare IgG and IgM, then affinity of IgM is lower for the antigen as compared to IgG antibody. But now if you consider the number of interactions, then IgG can uh, interact at only two sites with the antigen molecules, but IgM can perform 10 interactions with the multiple epitopes present on the antigen. So what will happen as a result of that? When this IgM will interact with the first antigen or first epitope on the antigen molecule, then it will come closer to the other epitopes present on the same antigen, and due to that the probability of interaction between IgM and the other epitopes present on the antigen is increasing. So you can see that in figure that other epitopes present on the antigen, multivalent antigen, are also coming in position and they are forming an interaction with the IgM antibody. So this is the meaning of avidity molecule. The next characteristics of the antigen and antibody is the cross reactivity. Now, what do you mean by cross reactivity? Now, for example, if two antigens are present which are carrying the similar epitopes on their surface, okay, antigen A and antigen B, both are carrying similar epitopes on their surface, and antibody is secreted against the first antigen. Then what will happen? This antibody was secreted against the antigen epitope which is present on the A antigen. But due to the similarity in the structure of the epitopes, now this antibody cannot recognize whether it is attaching to antigen A or antigen B. And so it will go to the antigen B which is carrying the similar epitope on its surface. So this is called as cross reactivity, where antibody which is secreted against the first antigen will recognize the epitope on present on the second antigen, and it will go and attach to that second epitope or second antigen. So this cross reactivity occurs if there are two different antigens that share identical or very similar epitope. So antibody's affinity for the cross reacting epitope, that is for the second epitope, is usually less than that for the original epitope, that is first epitope for which it was basically synthesized. Then cross reactivity is often observed among the polysaccharide antigens. Why? Because polysaccharides are formed by attachment of the monosaccharide units, that is like glucose, galactose, are attached to each other to form longer structures. So what basically happens is the basic structure of monosaccharide unit is going to remain same in all these different polysaccharides, and due to that if antibody is secreted against the first type of polysaccharide, like two polysaccharides are formed by joining the glucose residues only. So if antibody is formed against the first polysaccharide, then what will happen? Glucose is present in the second polysaccharide also, so it can go and attach to the second polysaccharide also. So for understanding this, we will consider one example of ABO blood group antigens. So these are basically the blood group antigens which are present on the RBCs in your blood. So if A antigen is present, then your blood group is A. If B antigen is present, then your blood group is B. If both the antigens are present, then your blood group is AB. And if none of the antigen is present on the RBCs, then your blood group is O. So now we will see. Now we will see what is the if you are having a specific blood group. Like for example, if you are having A blood group, then what will happen is on your RBCs A antigen will be present, but in your serum anti-B antibodies are going to be present. So why this is so? The reason for this is the cross reactivity of the antibodies. So now we will see why this happens. So if you consider the structures of this antigens, then basically these antigens, that is blood group antigens, are the polysaccharides which are composed of different monosaccharide units which are joined to each other. So in this figure, what you can see, there is O antigen which is carrying terminal galactose and fucose residues. Now if on this O antigen N-acetyl galactosamine is added, that is you can see a red residue is added on this antigen, then it results into the formation of A antigen. And if instead of N-acetyl galactosamine galactose is attached, that is you can see in the B antigen. So this is the structure of B antigen. So basically, if on O antigen N-acetyl galactosamine is added, then it results into the formation of A antigen, and if galactose is added, then it results into the formation of the B antigen. So this is the basic biochemistry of your blood groups or blood group antigens. So now we will see how this affects the composition of your composition of antibodies which are present in your serum. So as I have told you, these three residues, that is N-acetyl galactosamine, galactose and fucose are present on the are present in the microbes also, and these microbes are present as a common gut microflora inside your intestine. So what will happen is, during the development of the immune system, your cells will get exposed to this microbial antigens and they will start synthesizing antibodies against all these three residues, that is N-acetyl galactose, the galactosamine, galactose and the fucose residue. So all these three antibodies will be synthesized. But now I have told you that what is the composition of your blood group antigens. So what you can see in this figure is N-acetyl galactosamine is present in A antigen also, and it is present on microbe also. Then if you consider fucose, then it is present in all the three antigens and it is present on microbe also, and galactose is present in the B antigen and it is present on microbe again. So what happens now? See, if my blood group is A, then what will happen? My RBCs are going to carry A antigen on its surface. That means they are going to carry terminal N-acetyl galactosamine and fucose residue on the surface. So what will happen is, the antibodies which are synthesized against these antigens, if they are present, then what will happen? They will target my own RBCs, and due to that my RBCs will get destroyed. So to avoid that, what will happen is, the antibodies against the A blood group antigens will be eliminated, and which antibody will survive? Only the antibody against the B antigen. So what is happening here? My blood group is A. Okay. So due to the A blood group, on the surface of RBCs, N-acetyl galactosamine and fucose are present. But these are my self antigens. So antibodies against these two residues, that is N-acetyl galactosamine and fucose, are eliminated from my body, and what is remaining? Only the antibody against the B antigen. So here you can see that A blood group is present. So on this surface N-acetyl galactosamine is present and fucose is present. So these two are present as the self antigens, and due to that antibodies against these two antigens are eliminated and only anti-B is present in the serum. Now if my blood group is B, then what will be present on the surface of RBCs? Similar to A, galactose residue will be present and fucose will be present. So now these two are my self antigens. So antibodies against these two, that is anti-B and antibody against fucose, will be eliminated, and in my blood only anti-A will be present. Now if my blood group is O, then as I have told you, both the antigens are absent on the surface, that is neither A or nor B is present on the surface of RBCs. So what will happen? Only the antibody against fucose will be eliminated, and both the antibodies, that is anti-A and anti-B, will remain in the serum. And now if you consider last type of blood group, that is AB, then it carries both the types of antigens, A as well as B. So all the three residues, that is N-acetyl galactosamine, galactose and fucose, are present on the surface of RBCs. So all the three antibodies are eliminated and none of the antibody will be present in my serum. So why this is so? Why these antibodies are eliminated? Because these antibodies, even though if they are secreted against the microbial antigens, they can go and interact with the antigens, blood group antigens present on the RBCs, and if they interact with these antigens, then they will elicit the immune response, activate the immune response, and due to that this RBCs, your RBCs, will get destroyed. So this reaction between the uh, this reaction between the RBCs which are present on the donor's RBCs and the antibodies which is present in your serum is called as the blood transfusion reaction, which is basically a type of cross reaction only. So when blood is received in a transfusion, it must have the same antigens as yours, that is, it should be compatible. Both donor and receiver should carry the same antigen on their surface. If you get incompatible blood, that means if you get a blood transfusion that has different antigens, then what will happen? Antibodies which are present in your plasma will destroy the donor's blood cells, and due to that blood clotting will be there and it can cause severe effects on your body. So this cross reaction between antigens on the RBCs of the donor and the antibodies which are present in the plasma of recipient is called as the transfusion reaction. So this is all about the characteristics of the antigen and antibody interaction. Now we are moving to the next concept, that is lattice hypothesis. So when this antigen and antibody interact with each other, what happens is immune complexes are formed. So the ordered immune complexes which are formed or aggregates of these immune complexes are called as lattices, and these are basically formed by cross-linking between the antigen and antibody molecule. So for the formation of this lattice, the antigen and antibody must be bivalent or polyvalent. So what is required here? If you consider this antibody, you can see that it is interacting with multiple antigens. That means that antibody is bivalent. On the other side, when you see this in the second diagram, when you see that antigen molecule, it is interacting with the two antibody molecules. So it is again bivalent only. So if such type of multiple interactions or multivalent interactions are occurring between antigen and antibody, then one antibody can interact with two antigens. Similarly, one antigen can interact with two antibodies, and this can result into the larger aggregate or larger complex formations, which is called as lattice. So in this figure, you can see that there are antibodies and antigens present. So when they are interacting with each other, they are forming a cross link, and due to that, a larger complex or aggregate is formed, which is called as lattice, and their formation is called as lattice hypothesis. And due to this lattice hypothesis, it is possible to visualize this antigen and antibody interactions by precipitation or agglutination. So precipitation occurs when soluble antigen is there, and agglutination occurs where particulate antigen is present. So next concept that we are going to discuss is the zone phenomenon. Now when you consider a precipitating curve, that means a curve on which you take a specific quantity of antibodies and you go on adding the antigen molecules. So what happens in this is, at a particular time the concentration of antigen and antibody will be equal, and this is the point where there will be proper cross linking between the two molecules, and it will result in the immune complex or aggregate formation. Now before that point and after that point, no precipitate will occur because either antigen is present in the excess or antibody is present in the excess. So the point where the or the region of the precipitin curve where the antigen and antibody concentrations are optimal or equal for the lattice formation is called as the zone of equivalence in the precipitin curve. Now on the either side of this equivalence zone, what will happen? Either excess antigen will be present or excess antibody will be present, and due to that precipitation or lattice formation will not occur. So these two zones are called as pro-zone and post-zone. So this we will discuss by using a diagram. So you can see this curve. On this curve, on x-axis you can see antigen is added, and on y-axis you can see the amount of precipitate which is formed in the reaction is given. So you can divide this curve in three segments. In the middle region it is zone of equivalence. On the left side there is pro-zone, that is excess antibody region, and on the right side there is post-zone, that is excess antigen region. So in all the three zones, you can see that the number of antibody molecules which is present is equal. That is, in this diagram there are 5 antibody molecules present. So in the first region of the curve, what is happening? The number of antigens is less. So as compared to number of antigens present, the antigen binding sites which are available are more, and due to that what is happening? Individually this antigen is getting attached to different antibody molecules, and due to that no cross linking is occurring. So in this region precipitation has not occurred, cross linking has not occurred, and due to that lattice is not formed. So this region is called as prozone, and inhibition of the precipitation due to the excess of antibody present in this region is called as the prozone effect. In the third region, on extreme right, what you can see is there is post zone. Now what do you mean by post zone? In this region there are more number of antigens present as compared to antibody. Antibody concentration is same, that is 5 antibodies are present, but the number of antigens has increased, and due to that what has happened? Individually they are occupying all the antigen binding sites present on the antibodies, and there is no cross linking or sharing of antibodies present between this antigens. And again, no cross linking means no precipitate form, no lattice formation, and due to that here also in this region also precipitation is inhibited, but here it is due to the excess of antigen molecules which are present, and this region is called as post zone, and this inhibition effect is called as post zone phenomenon. Now in the middle region of this curve, what is happening? Both antigen and antibody are present in equal concentration or optimal concentration, and due to that there is proper cross linking, and due to that immune complex larger aggregate is formed, and it is resulting in the formation of precipitate. So how you can see the concentration of precipitate? In the first region it is lower, it is increasing in the zone of equivalence, and again it is declining in the post zone region. So where you will get maximum interaction between antigen and antibody? In the zone of equivalence. And when you are performing diagnostic tests for detecting these interactions, there you can see this formation of lattice in the form of agglutination or in the form of precipitation in the zone of equivalence only. That is when optimal concentrations of both antigen and antibody are present. So this is the meaning of zone phenomenon, pro-zone phenomenon and the post-zone phenomenon. So today what we have seen is what are the antigen and antibody interactions, which are the forces involved. So based on this, students, you need to attempt these three questions to clarify your concepts. So this is your assignment for today's session. Now we will take a brief summary of today's session. So we have seen today that antibodies are specific against the antigenic determinants. That means if complementary structure is present, then only they will interact with it and recognize that antigen and mark it for the elimination. Then we have seen this antibodies can cross react. That means if two antigens are present which are carrying the similar epitopes on their surface, then antibodies secreted against the first antigen can interact with the second antigen also. This is called as cross reaction. We have discussed this by taking the example of ABO blood group antigen. Then we have seen the concept of lattice hypothesis that describes how immune complex is formed in the zone of equivalence. And then we have seen what is proton effect, that is, it is the inhibition of the precipitation due to the excess of antibodies present in the first region of the precipitin curve. So this is all about the today's session. Now in the next session we are going to discuss about the different types of the diagnostic tests which can be used for detecting these interactions between antigen and antibody. So thank you for your attention.